Although Alfred Sturtevant was only 19 years old, he was one of Thomas Morgan's most promising assistants in the fly room. Sturtevant hypothesized that genes are located on chromosomes, much like beads on a string. The closer any two genes are on the same chromosome, the less likely they will be separated during crossing over. Conversely, the further apart two genes are, the more likely it is they'll cross over. By measuring the frequency that genes crossed over, Sturtevant constructed maps showing the relative position of genes on the chromosomes. He assigned map units based on how often genes crossed over. For example, if two genes crossed over 25% of the time, he said that they were 25 map units apart. Let's take a closer look at how this works. Suppose we know that genes A, B, and C are located on the same chromosome. The frequency of crossing over is 30% for A and B, 3% for B and C, and 33% for A and C. Can you determine what order the genes are in on this chromosome? According to Sturtevant, if genes A and B cross over 30% of the time, they're 30 map units apart. B and C cross over 3% of the time, so they're 3 map units apart. We can quickly see that A and B are far apart, and B and C are close together. Now comes the tricky part. In what order do these genes occur? Is it ABC or ACB? Again, if we follow Sturtevant's work, we can figure out the answer. The distance from A to C is 33 map units. Can you figure out the right choice now? Alfred Sturtevant did his groundbreaking work on the fruit fly, Drosophila, meticulously mapping the location of their genes. This modern map shows the location of genes on one of Drosophila's 4 pairs of chromosomes. The numbers indicate distances determined from crossing over data. Today, geneticists use much more sophisticated gene mapping techniques, such as radioactive labeling and gel electrophoresis. These techniques and others are currently being used to map the human genome, that vast collection of genetic material that forms the basic human blueprint from which each of us is built. Scientists with the Human Genome Project have already located genes for Alzheimer's disease and some forms of cancer. Perhaps one day we will be able to use this information to prevent or correct many genetic disorders.
Chromosomal gene mapping has allowed us to understand the genetics of inheritance in a fantastic way. Originally, many years ago, we identified what the chromosomal makeup of our cells are. And each individual has one set of chromosomes that we inherited from our mother and one set of chromosomes that we inherited from our father. So in that particular case, we are like carrying 2 sets of Britannica encyclopedia in each of our cells. And chromosomal gene mapping has allowed us to identify exactly where on the chromosomes a particular trait is, whether it be blue eyes or whether it be the gene for insulin associated with diabetes. And in the last few years, we've been able to map many, many genes on our chromosomes. And because we can map them, we can now understand a little bit about the basic cell physiology and genetics that makes me, me, and you, you.
In 1947, American geneticist Barbara McClintock hypothesized that small fragments of genetic material could move from one position to another on the same chromosome. Although most of the scientific community dismissed her idea, McClintock persevered with her research on jumping genes. It took 20 years before the scientific community accepted her ideas, but she eventually received a Nobel Prize for her work in 1983.